and welcome to the Absolute Recap Biology Edition. I'm your host, Melanie Kingett, and I'll be your guide to scoring a 5. Here at the Absolute Recap, we aim to maximize your understanding and minimize your need for memorization. Each episode will review content, skills, and test-taking tips to help you succeed in May. Your recap starts now. Hi, and welcome to the Absolute Recap Biology Edition. Today's episode will recap DNA replication. Let's zoom out. We're in Unit 6, Gene Expression and Regulation. Topic 6.2. Our big idea is information storage and transmission. Sperm and egg came together at fertilization, and the diploid number was restored. 46 chromosomes. Ever since then, the cell has copied that original DNA strand over and over again as the organism grew and differentiated. Imagine copying something that is 3 billion letters long without making a mistake? Cells aren't perfect, but they've got a few tricks up their sleeve. Let's zoom in. Recall that eukaryotic cells go through phases of the cell cycle. Interphase with G1, S, and G2, before cellular division with mitosis and cytokinesis. The S of interphase stands for synthesis, the time when DNA, in loose chromatin form, will be replicated with the assistance of enzymes. This process occurs in the nucleus of the cell. A quick pause to connect to the big idea. How does the structure of DNA influence the process of replication? DNA is an anti-parallel double helix, with the sugar phosphate backbone and nitrogenous bases joined through hydrogen bonds. The meat and potatoes of DNA is the sequence of the bases, the A's, T's, C's, and G's. In order to replicate the sequence, we will need to break the hydrogen bonds to access the code. Additionally, DNA has directionality, and just like the English language is read from the left side of the page to the right, enzymes will interact with DNA in a singular direction. Listen back to episode 17 for a chemical recap of DNA directionality. While there are several enzymes and intermediate steps of DNA replication, not all are specified in the CED. Let's approach the process of DNA replication through the function of these enzymes. First up, topoisomerase. This enzyme relaxes the DNA supercoiling so that hydrogen bonds are more easily accessible. Next, the enzyme helicase unwinds the DNA, breaking the hydrogen bonds between nitrogenous base pairs and forming a replication fork. Now that the nitrogenous bases are exposed, semiconservative replication can begin. Each original half of the parent strand serves as a template for new complementary nucleotides to be synthesized. In other words, half of the original strand is conserved or saved during replication. A short RNA primer is added to the template strands, indicating and supplying a replication starting point for our next enzyme, DNA polymerase. Enzymes are not named very creatively, and so literally this enzyme is involved in making a large polymer of DNA. DNA polymerase synthesizes the new strand in the 5' to 3' direction, laying down new complementary nucleotides and proofreading its own work as it goes. On the leading strand, this synthesis is continuous, in one piece, as DNA polymerase follows in the same direction that helicase unwinds. But remember, DNA strands are anti-parallel, so DNA polymerase is synthesizing away from the direction of helicase on the, very appropriately named, lagging strand. This causes the lagging strand to be synthesized in short pieces, named Okazaki fragments, after the husband and wife team that discovered them. We are almost done.
Hooray!
Quick enzyme check-in. Topoisomerase unwound, helicase unzipped, and DNA polymerase added new nucleotides. The last enzyme up to bat is ligase, the glue of the operation. Ligase will seal any gaps left behind by the now removed RNA primers and ensures that Okazaki fragments are joined on the lagging strand. Since DNA is organized differently in prokaryotes and eukaryotes, it should be no surprise that the replication patterns have variation as well. Prokaryotic DNA is organized in a singular, circular chromosome and has only one origin site of replication. Eukaryotic chromosomes are linear, contain multiple origin sites of replication, and often lose the ends of their chromosomes, called telomeres, with each replication. The shortening of telomeres is believed to influence the process of aging. To recap, DNA replication is semiconservative and occurs before the process of cellular division. A series of enzymes unwind, read, and synthesize complementary strands while accommodating for antiparallelism. Today's question of the day is about cell division. What is the condensed, identical DNA strand called during mitosis? For the answer to today's and future questions, please follow us on Instagram at TheApsoluteRecap. That's the APsolute recap. If you are a student with questions or a teacher with suggestions, we'd love to hear from you. If you have a biology topic or another AP subject you would like us to cover on the Absolute Recap, please email us at TheApsoluteRecap@gmail.com. That's the APsolute recap@gmail.com. The Absolute Recap is produced by Brad Kingett with music by Zach Caruso. Please remember to rate, review, and subscribe wherever you get podcasts. Time's up, pencils down. Thank you for listening to the Absolute Recap Biology Edition. AP is a registered trademark of the College Board. Copyright 2020, Absolute Recap, LLC, all rights reserved. Coming up next on the Absolute Recap Biology Edition, Unit 6, Central Dogma.